Plants. We're going to totally shift gears now to talk about from rare plant and seed collection to mapping of vernal pool ecosystems or updated mapping. So online here is Regan Murray. Regan has a BS in Geographic Information Systems from the University of Wisconsin-Madison. She has worked as a GIS analysis for the Bureau of Land Management Plant Conservation and Restoration Program and is now with the San Francisco Estuary Institute, where she does research and development of automated mapping methodologies to identify and classify wetland habitats and potential pollution sources of contaminants of emerging concern across the state of California. So with that, take it away, Regan.
Thanks, Russ. Thanks so much for the introduction, and thank you so much for letting me present. I know we're kind of nearing the end of the conference here, so folks are probably pretty tired, but I have a big smile on my face because I am so stoked to be here. I grew up in the East Bay of the Bay Area, hiking my dogs in the Shell Ridge open space past vernal pools every single day. So to be giving a presentation on vernal pools to some of my fellow plant nerds is just like a huge, awesome full circle moment for me. So I'm really excited to get into it. We're going to be talking about some mapping methods for mapping individual vernal pools across the Central Valley of California. A quick overview of what we're going to be talking about. We'll go over a little bit of the ecological context of vernal pools. We'll talk about the methods I use to map these pools across the Central Valley, future work, and I'll pair you up with some resources to learn more about vernal pools. All right, so what are vernal pools? They're not exactly the poster child of California habitats. They're not very glamorous, but what they are is ephemeral wetlands formed when rainwater pools in small depressions over an impervious soil layer. So what does that mean? They're basically really shallow depressions in the landscape with a really firm layer of soil beneath, usually clay or actually bedrock, that prevents water from leaking into the soil. So it creates these shallow pools. And these pools are unique and they're different from ponds because they actually have a really short phenology. And what you see here is kind of like the cycle of vernal pool phenology. For a really short period in the springtime, around March or April, they're in this aquatic/wetting phase. And this phase only lasts for a few weeks, at most a few months, if it's a really wet season, before they move into this flowering phase. As the water doesn't run off at all, it relies on evaporation to kind of enter this flowering phase, and eventually it completely dries out. And most of the year, it looks like this picture in the bottom right, the drought phase. But this is really what makes vernal pools unique. They have this kind of exaggerated phenology that occurs in a really short period of time. And what that allows for is some really interesting and unique species to make their homes in these places. So it is a critical breeding habitat. Animals like the California tiger salamander, the vernal pool fairy shrimp, the vernal pool tadpole shrimp all breed in these pools because they're aquatic environments. But what that short phenology allows for is there's no fish in these places. So predatory fish that would traditionally eat the eggs of salamanders, shrimp, etc., don't really have the chance to prosper in these places, which gives these prey animals a chance to really establish their own communities here. Additionally, kind of the juicy topic for all of our botanists, they're huge supporters of native plant communities. There are over 200 California natives that call vernal pools home. And of those 200 species that you can find in a vernal pool, 100 of them are endemic to vernal pools. So really special habitat. And out of those 200 species that occur in vernal pools, over 30 of them are state or federally listed as endangered. And we'll kind of get into the reasons behind that in a few slides here. But beyond being incredible habitats and supporting all of these wonderful plant and animal communities, they also provide great erosion control and water filtration for what is often the surrounding wetland habitat. So these are just a few of the faces you might find if you had a magnifying glass and you're at a vernal pool. The turtle obviously would be a little bit bigger, but the turtles are actually kind of restricted to the East Coast vernal pools. In the California vernal pools, you would see these fairy shrimp and these tadpole shrimp. But I know what we're all really here to see are the superstars, the California natives that call vernal pools home. Like I said earlier, there's over 200 species that call vernal pools home. This is just 6 of them. I believe all of these 6 are state or federally listed as endangered. someone has to fact check me on that. But quickly, I'm no botanist. I just wanted to share some fun facts about some of these plants. All of these plants that live in our endemic vernal pools are true California natives. Like I said, they're only seeing water for about 2 or 3 weeks throughout the year. So they're extremely drought tolerant, which, given the circumstances in the coming years, is a really valuable trait to have in plant communities. Additionally, they have some interesting habits, like a lot of them have long, hollow stems that draw air to the roots through the vernal pool. They can pull atmospheric gases directly from the water using specially adapted leaves. Um, so lots of good stuff that these uniquely adapted California plants can achieve. But I talked about earlier, there are over 30 threatened plant or animal species that are endemic to vernal pools. And the reason for that is, as of 1976, 90% of vernal pool habitat in California has been destroyed. And on top of that, in 2005, Bob Holland did a study that determined that of that remaining 10% habitat, 13% of that habitat had been destroyed. So I'm not a mathematician. I can't on the fly tell you what remains of that original 10%, but it continued to decline to this day. And the primary reason for that is agricultural expansion. The California Central Valley is famous for its agriculture, and that's the primary reason. Secondary reasons include urban development and man-made changes in hydrology. So the digging of canals might redirect water that would pool in these like vernal pool depressions to other places. And here's just a picture of what that might look like. So this is an example that comes from directly east of Santa Cruz near a town called Firebaugh. You can see this was kind of a huge vernal pool complex in 1998, but we can since see that it has been plowed over and turned into a large agriculture field. And you can kind of see, I think it's interesting the way the landscape kind of carries the memory of the system. You can kind of see like the watermarks across the agriculture, which is neat. But this is the primary threat to vernal pools and why it's so important that we keep track of where they are. So vernal pools are facing this challenge where we have a very limited amount of habitat left of vernal pools, and all of these species rely on them to survive. So we want to restore and conserve these areas, but our knowledge of the on-the-ground conditions is limited by data availability. And kind of up until last year, the best available resource for identifying vernal pools is what you see on the right-hand side of the screen here. And this is still great. These are the Carroll-Witham Vernal Pool Complex polygons, and basically they delineate vernal pool habitats. So the idea behind these is like within these polygons, there are vernal pools, or there could be vernal pools. But beyond that, that's kind of the extent of the information we have on vernal pools, which is helpful, but can make it hard to make strategic management decisions or landscape planning decisions. So the goal of this project was to resolve that and fill in those data gaps by mapping individual vernal pools within those Carroll-Witham Vernal Pool Complex polygons we were looking at on the previous slide. So we did this using an object-based machine learning model, which I know is kind of word soup. We'll talk more about that in a minute. And we were able to map around 83% of the vernal pool habitat in the Central Valley, which is really exciting. So I know this is total word soup, but our approach was doing classification with an object-based random forest model trained on over 1,100 points using eCognition. So this is probably pretty meaningless to you right now, but maybe we can go through it step by step and understand exactly what was going on here. So we started by developing some classified training points. We derived image objects from LiDAR fill difference. We trained a random forest model, and we applied a random forest model. Our data inputs are shown here. We use high resolution LiDAR from the 3D elevation program from USGS, and as well as high resolution imagery from NAPE 2020. Both of these are free resources, which is great if anyone's interested in attempting to recreate these methods. It's all available for free through USGS and NAPE. And as you can kind of see on the right-hand side, this is what our input data looked like. And on the top right is what I think you should pay attention to in particular. This is the LiDAR fill difference, which is just a derivative of LiDAR data that shows depressions in the landscape. So all of those white spots you're seeing are small depressions in the landscape. And those are all vernal pools. So you can see how well this data highlights the vernal pools that we're trying to map. So again, we started out by developing classified training points. We had a trained wetland scientist at SFEI dropped all of these points. There was 1,100, and we used a binary system. So either it is a vernal pool or it is not a vernal pool. So you can see here, for example, all of these point data dropped in ArcGIS Pro inside of these vernal pools and labeled as vernal pools. The next step was deriving image objects from the LiDAR fill difference. So on the top, you can see the LiDAR fill difference. Like I mentioned, this is showing all of those small depressions that represent vernal pools. And on the bottom are the image objects we were able to derive from there. So you can kind of see how the red outlines correspond almost directly to those LiDAR objects. So we just use this LiDAR fill difference to produce objects. And with that, we paired those objects with the training points. So basically we have this polygon that we derived from the LiDAR fill difference. And now we put this point on top and say, this is a vernal pool or this is not a vernal pool. And that left us with the final step and also leads us to the good news. We applied the random forest model across the Carroll-Witham polygons and got to the good news, which is that we now have individual vernal pool data across the Central Valley. And I'll go through some closer up examples so we can kind of see the impact of how much more specific mapping the individual pools is. So this was kind of the way the data existed before. We have these large habitat polygons that have since been refined. You can now see in yellow, we can see the individual vernal pools mapped within the complex polygons, which is really exciting. It's not perfect, but it's a great start and more detail than was available beforehand. I'll just click you through a couple more examples. You can see it pulled out those little depressions. Here as well, we've seen this picture. This is at Jepson Prairie, which is a UC Davis research reserve, I believe. And here are some from Southern California, so maybe not as interesting, but on the top right, those are some man-made vernal pools. So interesting to see how the method can work with different types of vernal pools. So the results, we were able to map 675,000 acres of the Central Valley vernal pool habitat, or 83% of the Witham complex polygons. That lingering 17% that we weren't able to map comes from just data gaps in LiDAR data availability. So there wasn't data covering 17% of the habitat, so we weren't able to map in those spots. We tried a few things to get at those areas, but without the LiDAR data, it was just kind of difficult to get there. But in the future, it's something we would like to fill in. I've seen that USGS is planning on flying LiDAR in those areas. So hopefully down the line, the model can be reapplied and we can fill in those data gaps. Just quickly, I wanted to cover some shortcomings in the model. So like I mentioned before, the data gaps leading to that 17% area not mapped. Additionally, this method really only works within those predefined Carroll-Witham complex polygons. So you could not just drop this across the whole state of California and it would find all of the individual vernal pools. You kind of need to give it a bounding box of a vernal pool habitat already. Additionally, there was a bit of trouble using the single date imagery. As you can see on the right-hand side, that image is very low contrast. You can see these areas get really dry and it's really difficult to discern where the pools are. So it would be great in the future to introduce some time series imagery or imagery taken during the wet season. And this again just kind of illustrates those issues with, on the left-hand side, some low resolution data, and on the right-hand side kind of showing that nape imagery on the top that's really dry and how it kind of misses some of those key features that are shown when there's a lot of water on the ground. In the future, we would love to make improvements to mapping both individual vernal pools and complex systems. I don't really want to bore you all with the specific details, but on a high level, we would love to introduce some time series imagery, so imagery from throughout the year, particularly the wet season, so in March or April, to capture when the pools actually have water in them. They're a lot easier to see. Additionally, we have ideas for other LiDAR derivatives and data sets we could use to improve identification of complexes and systems, which would be really exciting. There's a lot of opportunity to learn about vernal pools in California and to get involved. This data can be downloaded from the SFEI Data Center website. I've linked both of those here, but you can also go and visit vernal pools in person across California. There's Jepson Prairie, the Prairie City State Vehicle Recreation Area, Phoenix Park, and Illa M. Collin Conservation Preserve. Those are all in Sacramento, so y'all are lucky with the good vernal pool access. Additionally, there's a great free ebook about conserving vernal pools in California written by John Volmar, and I've linked that here as well. And if you do decide to go see the vernal pools in person, I found these really fun vernal pool wildflower reference guides. This is just 2 screenshots, but they have a bunch of content that you can print out or pull it up on your phone and bring with you when you're walking around all of these awesome parks. So thanks so much for listening, everyone. I'm happy to take questions. I think we have a little bit more time.
Great. We've definitely got time for questions. Any questions for Regan? Got one in the middle.
Hi, wondering if you were able to test the accuracy of the classification, or if you know what the accuracy was. And also, did you need to manipulate the aerial imagery at all to, I don't know, to get it to be consistent across like a large area, or to better show the pools or anything like that?
That's a really great question. We unfortunately, due to funding and time limitations, did not do an accuracy assessment for this project. Although I think if we ever received additional funding, that would be the next step for this project for sure. So that's a very good question. And did we do anything to the imagery? No, we didn't. And I will say that the model was highly reliant on the LiDAR data. It actually barely used the imagery because, like I showed, we had that really, all of the nape in California is flown at the same time of year. It's flown at the end of May, which for the Central Valley is already pretty dry. So all of the imagery was kind of useless, to be honest. So we didn't do any manipulation to standardize the imagery across the state. But I think if we eventually get to use that planet imagery, like I'm showing on the upper right-hand side, we would definitely do some of that because I imagine with the right imagery, it would play a much bigger role in the model.
Did you happen to do any field verification of your data set?
For this project, for the Central Valley, no, but we did do field verification for this mapping that you're seeing on screen is in San Diego, and we did do field verification for the vernal pools in San Diego, which was a separate project technically from the Central Valley, but using the same model.
I had another question about how you used the LiDAR, which was really interesting. And I think you mentioned at the beginning that vernal pools are on areas that the water doesn't infiltrate into the ground. So I was wondering if the main way you're identifying them is just looking for the low points in the ground. How do you make sure that it really is an impermeable area?
Yeah, that's a really great question and a really good point. So the reason that this worked here is because we limited the model to areas that were confirmed vernal pool habitat by those Carroll-Witham polygons. So we had already confirmed that the circumstances were right for it to be a vernal pool habitat. However, this would not work if you didn't already know that. So that's something you need to know ahead of time and kind of need to limit the study area to a confirmed vernal pool habitat with that clay soil or bedrock.
Okay, we have time for one more question.
Hi there, thank you. That's a really interesting methodology. I was curious if you could speak more to the kinds of impacts that this more fine-scale mapping has, or like could have. You mentioned kind of like strategic management decisions and things like that. I'm just curious if you could expand on that a little bit more. Thank you.
Yeah, totally. I can maybe speak to actually who funded this project. So this project was paid for by Caltrans. They requested this data. The way that they used it was they actually took a bunch of those endangered species that I mentioned, over 30. They used all of this to kind of pinpoint exactly where those endangered species were living. So they were looking at like the vernal pools, fairy shrimp, the vernal pool tadpole shrimp, et cetera. And they were doing that for transportation infrastructure planning purposes. And how exactly they used the data, I'm not sure, but I know they used it to refine their endangered species mapping.
Well, great. Thank you very much, Regan. We'll appreciate it.